think he summed it up well there. Well, I'm Shron, and as Brian said, I work with the Discovery Consortium, which is being funded by the Emer Foundation, but I also chair the medical panel for Ovacare. So who are Discovery? Well, it's a consortium of researchers. It's led by Professor John O'Leary in TCD, but it consists of gynecologists, oncologists, pathologists, scientists, bioinformaticians, as Juliette was also telling you about, and from universities all across Ireland, but also we have links with universities across the US as well. And the Discovery Consortium, I suppose one of the important pieces of infrastructure that we have is this a bioresource where we recruit patients coming in for surgery, and all patients are asked to give a blood sample and a tissue sample for research. And this is a massive bioresource for any of the studies that we do in ovarian cancer. And as Juliette already told you about the Emer Foundation, it was set up in memory of Emer, who died from ovarian cancer, and the foundation's funding 3 PhD studentships in Trinity College. And I'll talk to you a bit more about their work as we go on, but Mairead Murphy and Linda McAvoy have successfully completed their PhDs, and Brendan French should be submitting his in December. Mairead just had her PhD viva this week, and she has very encouraging work coming out of this. So I suppose as scientists, one of there's 2 major challenges that we see in ovarian cancer. And as you heard from Sheila earlier this morning, early stage disease, which has a good prognosis, cannot be detected easily. And there's a lot of vague symptoms, and as you heard from Sheila, it's also often referred to as the disease that whispers. But also some of the standard chemotherapy approaches often fail, and patients develop recurrent chemo-resistant disease that no longer responds to chemotherapy. So as scientists, these were some of the major challenges that we wanted to focus on in our work. So currently for diagnosis of ovarian cancer, a GP can perform an internal exam. There's also a blood test, and you heard earlier about CA125, the tumour marker has been mentioned. However, CA125, not all tumours will express CA125. So it's not it's sensitive, but it can be very non-specific in the diagnosis of ovarian cancer. And then pelvic examination is often carried out by a gynecologist, and then the patient might be referred for an ultrasound. Now, CA125 is the only tumour marker that's out there at the moment for ovarian cancer. It was discovered back in 1981, but there's about 20% of ovarian cancers that don't express it. And it can also be elevated in a lot of non-cancerous conditions. And there's other biomarkers coming out there at the moment now. One of these is HE4, which I'll tell you a little bit about later. So for screening, currently there's no standardised reliable screening test for ovarian cancer. As I said, pelvic examination is widely practiced, but it's not sensitive enough to be used as a reliable screening tool. And CA125 can be found also in non-cancerous conditions, so it's not a very good marker for screening. Now, there was a recent trial done in the US looking at screening for ovarian cancer, and what they found was that screening didn't actually improve the outcome of women. It didn't make, having screened patients, it didn't make them survive longer. Now, there is another trial which is using the same ultrasound and CA125 for screening, and that's going on in the UK at the moment. And what's different about this trial is that they're looking at how your CA125, because a lot of patients might have normal CA125s, but they're looking at the change of CA125 over time as opposed to just a cut-off value. Now, the screening for this finished in 2011, but there won't be any results until it'll be December 2014 before we know whether this screening trial has shown beneficial results. So clearly, novel biomarkers are needed in the management of ovarian cancer. And I'm going to talk to you about 4 different areas which the Discovery Consortium has focused on at the moment. Autoantibody profiling, HE4, microRNAs, and circulating tumour cells. So apologies if I'm going to bombard you with a bit of science, but it's hard to avoid it as well in these talks. So this is work done by Mairead Murphy, who is the PhD student that just completed her viva this week. And it was looking at developing and validating diagnostic serum-based biomarkers in ovarian cancer. And Mairead used a technology called autoantibody profiling, and this was done in conjunction with Professor Dolores Cahill in UCD, who's also here today if anyone wants to know more about this, where she screened serum samples from healthy controls and compared this with serum samples from ovarian cancer patients. And this is just a typical layout or a result from one of these, where we see these spots lighting up here where there was an autoantibody profile detected. And what we found, I suppose, is that in early stage ovarian cancer, there are many of these autoantibodies detectable as opposed to late stage cancer. So really, early stage offers a good opportunity with this particular technology. So currently we have a panel of biomarkers which we are carrying out further validation on. And the other area is microRNA profiles. Now these are little molecules which can be found in the blood. They're all over the body, but they can regulate your gene expression. And previously these were thought to be part of what they call junk DNA. But now, as I suppose as science technology has evolved, they have found that these are actually very important for regulating gene expression. Now, these are attractive markers because they can be isolated from the blood and they're quite stable. So we are currently looking at these microRNA profiles in patients. The other marker, which is HE4. HE4 is a novel biomarker, and it's received FDA approval in the US, and it's found to be more sensitive and specific serum biomarker for ovarian cancer, and especially in premenopausal women. So we were in touch with the company that make this HE4, and we decided to evaluate it on our biobank of samples, which we have in Trinity College. Now, we looked at bloods from patients before they had their surgery. We looked at benign patients. We looked at patients who had what they call borderline disease. And we also looked at malignant patients. And we looked at these in two different cohorts, premenopausal and postmenopausal women. And what we found was that indeed it does improve the sensitivity and specificity when it's used in conjunction with CA125, the current marker that's out there. So there's a lot of sometimes false positives and false negatives with CA125, but this marker does improve this. So we're about to present this to the clinicians to show them the data, but it's encouraging to see that there might be another marker that might be useful in the diagnosis of ovarian cancer. And what we also found was that in some patients, when we looked at this marker before they had their surgery, and then we looked at the marker a couple of days after the surgery, and in most patients, this marker dropped off quite quickly, and as the tumour was removed, the marker dropped. Now, CA125 can do that also, but we did find that it stayed up in some patients, and these were often patients that went on to develop chemo-resistant or progressed, their disease progressed. So this was only done in about 30 patients, so we're going to carry out a larger trial on this marker, but it could be important to tell us which patients are more at risk of developing recurrent disease. Now, the other hot area, I would call it probably in the area of cancer in general, not just ovarian cancer, is the area of circulating tumour cells and the metastatic highway. So how do these cells spread around the body? So you can imagine you have your tumour. For it to spread around the body, it comes out of the primary site, it goes into the blood system, and then it develops in a secondary site. So how do these tumour cells go around the body without being taken out by your own immune system, and how do they avoid that? And one of the areas, I suppose, to try and find these tumour cells in the blood is like looking for a needle in a haystack, because they can be very rare, or at least current methodology thinks they are very rare. But what we have found out now is that a lot of these tumour cells, once they get into the bloodstream, they actually get cloaked in your platelets, which are in your own blood. So the immune system in the body can't recognise these because they're covered in platelets, and it can't recognise them as being foreign objects as such in the bloodstream. And what animal studies have shown in this is that if you take out platelets from animals, and if you introduce tumour cells, the ones that have no platelets, you don't get metastasis forming in those. And if you put in something to block the platelets, you get a lot less metastasis. So we now know, and it's become a hot area really in the area of science, is that platelets are very important. And this is just some of the work we have done where we've looked at how platelet cells interact with cancer cells. And if we just put them in a dish in the lab, they will just start to bind to each other. But if we introduce a force, which is called a shear force, and these are what the tumour cells experience as they're whizzing around in the body, then you see multiple platelets. They just really all attach to the tumour cells. And the other work that we have done is when we have looked at what is the platelet saying to the tumour cell once it sticks onto them. And really what it's saying is, do not die, keep surviving under any conditions. And these are the kind of signals that we've looked at the genetic level that the platelet is saying to the tumour once it sticks on there. So we've been fortunate with the Erie Casey Foundation as well to acquire a machine called a solid second generation sequencer. And what we want to do with this is to analyse these circulating tumour cells and find out exactly what is in there and to see, can we detect these in the lab? Can we pull out these cells? And we're using a technology, this was by a company in Paris, it's called screen cell technology. And it's a simple filter, because current methods for isolating circulating tumour cells use a marker on the surface of the cells called EpCAM. And from the work we have done, when the platelets are on top of the tumour cell, you can't recognise this EpCAM. So current methodologies are really underestimating the amount of tumour cells that are in the blood system. So this uses a simple filter technology where you literally put the blood sample through, and here it captures these tumour cells on the filter. So we can now take these and analyse them and see what exactly is composed in these tumour cells. And this just shows an example from the company that you can see it captures these tumour cells, but all these fuzzy bits down here at the end, these are actually platelets. So the theory is correct that these platelets are stuck to the tumour cells and have a really important role to play in the metastasis. And we can also take these filters and put them into mice, and what we find is that they generate a tumour. So we know that they are tumour cells that we are capturing on these filters. So currently we're working as part of a large consortium funded by Science Foundation Ireland to develop a new method for capturing these tumour cells. So I suppose that's the area of diagnosis and what we're trying to do in that area. The other big question, I suppose, ticket question in ovarian cancer is the area of chemoresistance. Like, why do these tumours grow back and why do they no longer respond to chemotherapy? So we've done a lot of work in this area, and I'll just talk about the few different projects. And one of these is where we compared tumours from women whose tumour grew back to primary tumours from patients. And we found a load of different markers that were under and overexpressed in the recurrent tumour. And one of these is a marker called MMP9. And what we did then in the lab is we got an inhibitor, something that would block MMP9. And then we just checked to see whether we could get the cells to respond to chemotherapy if this MMP9 was blocked. And indeed it did. So we've currently put this work in for publication. The other marker which we have been looking at is part of the inflammatory response. Now, ovarian disease is often, I suppose, it's known as an inflammatory disease as well, because there's a lot of cycles that the ovary is going through every month. There's a lot of inflammation going on there. And one of these markers is involved, called MYD88. And what we found was that we can detect this in tumour cells in ovarian cancer, and that the patients, I'll just go back one actually, the patients who have high MYD88 actually have a worse survival than the patients who have no or low MYD88. And also there's another marker which we've looked at in conjunction with our collaborators in UCD, and this is called MAD2. And what we found was if this is low in tumours, that the patients often have a worse outcome. So, sorry, no, I think we're missing some slides there, but I'll just go back. So where to next, I suppose, for all of these markers? Well, what we're currently doing is we're going to try to talk to ICORG about putting some of these markers into clinical trials, because you can't just find something in the lab and then introduce it into the clinic. They have to be rigorously tested and go through the clinical trial situation. So that's what we're currently at, but there's a lot of promise there, and some of the work that's been done on the cancer stem cells by our third Erie Casey student, we think that these are actually the cells that are responsible for the chemoresistance and for tumours growing back. So there's a lot of work, and I'd just like to thank Ovacare for giving me the opportunity to talk to you about this, and if anyone has any questions, we'd be happy to answer them after.